I'm really honored to be here today with Dr. Santosh Helekar, who is a neuroscientist at Houston Methodist Research Institute. He also happens to be an MD. He's a man of many talents and an incredible creative inventor of really world-changing things. He's also working as scientific director of translational biomagnetics. I have to work hard to say these things and make them come out. And professor of neuroscience research and psychiatry at Weill Cornell Medical College in New York. You are a busy man.
Yeah, trying to.
I'm grateful that you gave us some time today. So we met at the Science of Consciousness conference in Barcelona, where I was especially blown away with your presentation on so many different levels. I hardly even know where to begin. But I know I told you this already, but you were the only presenter there who spoke to issues of both the level of consciousness and the content. And you have tested for these things, which I find incredibly exciting. And the other thing you were the only person there who talked about was measuring something about consciousness away from the cranium. Like it's all about the brain. Everybody is all obsessed about the brain, of course, because there's a lot to the brain as a fascinating organ, but you actually are measuring consciousness on the hand, for instance, like away, far away from the brain. It's just fascinating. So yeah, I'm talking so much. Let me ask about this device, which is called a sentiometer, really measuring sentience.
Yep.
How did you come to that idea?
Yeah, basically, I've been thinking about consciousness for a long time. So almost since medical school, which was back in the early 80s for me. I graduated from medical school in 1982. And just out of medical school, I was interested in while in medical school, I was interested in knowing about consciousness, how it comes about, how the brain produces it, if it does. And I was disappointed with what they taught us in medical school, in that there was no known mechanism that anybody could explain of how people are conscious, but how consciousness emerged at all in evolution, right? And we all thought, having studied neurology and psychiatry, that the brain had to do something to do with it, of producing consciousness, but nobody had a clue as to what that might be. And my disappointment toward the end of my medical school turned into a kind of a drive, a motivation to study it, to conduct research, to understand it, rather than do clinical practice and see patients. And so that's how I began my career as a neuroscientist. I went to an institute back in India to do research, to understand consciousness, and actually started recording EEG from meditators as my first job.
Oh, wow.
Yeah. My first job was to record EEG from meditators.
Oh,
And
wow. That's very cool.
yeah, in a research setting and in a neurophysiology department at what's known as the National Institute of Mental Health and Neurosciences in Bangalore, in India. And then from there, I thought I should actually learn how to do scientific research, neuroscientific research. So I came to the US and got a PhD in neuroscience from Baylor College of Medicine. But all the while, I wanted to study the underlying neural or scientific basis of consciousness. And it wasn't possible back in late 80s and early 90s at that time, when I got my PhD and then continued my research and then got a faculty job at Baylor College of Medicine, because the question of consciousness was considered to be a philosophical question.
Yeah.
And to be studied only as a philosophical problem, and also maybe had this connotation of being something that is spooky and spiritual and not meant for scientific investigation. But in the 90s, people in the neuroscientific field became more interested in it and started thinking about it scientifically, like Sir Francis Crick, Christoph Koch, all these people started thinking about it. Gerald Edelman and Roger Penrose, who was a physicist, right? So Roger Penrose came to think about consciousness from a purely quantum physics standpoint. And he wrote a book called The Emperor's New Mind, which was very motivating to me. I was inspired by it to delve deeper into the question while I did my day job, trying to do bread and, as I said in my talk in Barcelona, do my bread and butter research in neuroscience. I was also thinking about this, and particularly thinking about, first of all, when I read Emperor's New Mind by Roger Penrose, I didn't think the main idea was testable. And the main idea was that each conscious moment is conversion of a wave into a particle deep inside the brain. And most likely it was electron wave into an electron particle, right? And in around 1994, I guess Stuart Hameroff became his collaborator and proposed that it was microtubules inside cells within the brain that might be responsible for where this collapse occurs of electrons potentially from waves to particles. And each such collapse, they thought, was a proto-conscious moment. And then a whole string of collapses then lead to more basic elementary conscious moments, which we call qualia, to be being philosophical about it. But that remained a theory for us, for everybody actually at that time. And I didn't think it would be testable ever. And they were talking about it, a lot of articles being written on it. But like in the middle of the 2010s, I guess, a new field was becoming born. It was called quantum biology because of some very interesting results, very interesting findings related to migratory birds that were flying from the North Pole, from the northern part of the hemisphere, down to the closer to the basically northern part of Africa during winter or during fall. And the question was, how are they migrating? And there were many theories. But one of the things that actually became clear was that they were probably using quantum mechanical effects on electrons, that in proteins, in specific proteins called cryptochromes in their retina, in their eyes, to map the magnetic field of the Earth. And that gave them directionality, yes, which direction to fly. And so a very weak quantum effect was used by these birds neurologically, and their brain, within their brain, to do some overt behavior, migratory behavior, which meant that perhaps there are other such quantum effects in the brain that might be important in biology. And then one of the things that immediately occurred to me and many other people is that perhaps consciousness, Roger Penrose is right, or in some way, that maybe quantum effects deep inside brain cells might be responsible for consciousness. And then from there on, I was working on the side with magnets, basically.
Yes.
To stimulate the brain, using what's called the transcranial magnetic stimulation technology. So we had miniaturized that technology to make it wearable so that you could stimulate multiple sides of the brain using magnetic stimulation. And the mechanisms that we were thinking about as to how these effects might be occurring were, again, probably something had some similarities with how the birds migrate down using Earth's
Wow.
magnetic field. And those that involves in electrons again. And it was becoming clear that perhaps I should concentrate on Roger Penrose's hypothesis and see if I can make that somehow testable. And one of the ways I thought we could, and this was pure speculation on my part, because again, I'm not a physicist. I could just basically expand if I were given free license to speculate, right?
Yeah.
Like a poetic license. I thought, say, what if this effect that Penrose is talking about inside microtubules, what if it spreads out through the brain and through the body and out into space? How could I detect such an effect? And that led me to think back to an experiment that was done 200 years ago by Thomas Young, which was what's called a double slit experiment, where you take a light source and you pass light through two slits, and then you get an interference pattern, right? And you get an interference pattern because the light is a wave. That's how Young determined that light, proved that light was a wave. But then in the 20th century, early part, the 1920s, they showed that if you knew which slit the light waves were passing through, or instead of getting an interference pattern, there was a tendency for light to form two bands instead of an interference pattern. And two bands are something that you would see if light was a particle.
Uh-huh.
Okay. So here was an apparatus that could allow you to distinguish between light as a wave or a particle. And so what we did was miniaturize that apparatus to detect whether this collapsing electrons or waves that come out of the brain, whether they collapse into particles. If the effect spreads out, the collapsing effect spreads out into the space and collapses light waves into particles, we would be able to detect that. And
So
that led to
is it biophotons that you're measuring?
This is not biophotons. Biophotons are actually produced in the brain, and people claim now that you can measure them in the dark, but this is not biophotons. It is just a quantum phenomenon. If it is true, it would be a new phenomenon that requires new physics, basically, which I'm not capable of working on, but I have collaborators now that are interested in doing that. But my idea was the collapsing electrons deep inside the brain have an influence on surrounding electrons. And it's like a domino effect. So a collapsing electron causes a neighboring electron wave to collapse. Okay. And then that causes its neighboring electron wave to collapse. And that keeps happening until it declines with distance, obviously falls off with distance, but it also spreads out in space, and it influences other wave functions, or like light wave, to collapse as well. And there are some more details that we are now thinking about, which I won't go into here because it involves a little bit of more explanation. But anyway, the bottom line is we made a miniaturized version of the Young's double slit apparatus. And we were able to measure the intensity of these light bands electronically, right? And then record those densities in multiple channels and derive a waveform as a result, which, if there was nobody in the room, you had this apparatus, and it was lying there in an empty room on the table. You saw a straight line. You didn't see any kind of deviations of the line. But if you brought this device close to the head of a person, you found that there was a nice large deviation in the baseline, which correlated with, we thought at that time, with the level of consciousness of that person. We showed subsequently that it was indeed that consciousness indeed had something to do with it, because if we did these recordings in a mouse and we anesthetized the mouse, the amplitude of the change that you see in this recording reduced as the
Yeah.
mouse became anesthetized. And as the mouse recovered from the anesthesia, the amplitude increased. Also, the time course showed a difference. So clearly, consciousness had something to do with it. And we have gone on now and done experiments or recordings in patients in intensive care units, patients who are unconscious and in different levels or at different depths of unconsciousness, either due to direct brain injury or impairment, or decreased level of consciousness because they were given anesthetics and sedatives. And we've found that most of these patients show a reduced amplitude of this response that we measure. We have simplified that device further in doing these recordings, because we found that we don't need to make the double slit apparatus to get this effect. We can study any kind of wave phenomenon, such as diffraction of light. And so diffraction of a laser light beam is the easiest way we can get waves to record intensity of light that is in the waveform. And if we show a decrease in that intensity of light as a wave, then that reflects we think its conversion into particle, or this process that causes light, the diffracted light intensity to decrease. And the most current device is actually, it just measures diffraction of light.
This is incredible.
Yeah. And so this is, so that's where we are doing lots of more recordings. We, as you mentioned, we not only does the level of consciousness modulate the recording level that we get when we record from these subjects, but we can also detect changes in content.
Yeah.
So, for instance, when somebody is focusing on playing a video game on the computer, right? So each time the person plays the game, we see a change in the amplitude that reflects
This is incredible.
the time during which the game is played. So it does. So mental activity determines, conscious mental activity seems to determine this response, what we call sentiometric response, right? So potentially this has, now, of course, we don't know whether the hypothesis that I tested, the very
Yeah.
speculative hypothesis, if that is the correct mechanism of the phenomenon or not. But we need to do other tests that have nothing to do with sentiometric recording to find out if what we are saying is correct or not. The mechanism of it is what we think it is, or in something else. But we find that these recordings are not just limited to measuring level of consciousness, but from people, you can generalize these recordings to other organisms as well. I told you already about mice. So we get the same kind of responses that we get from people in mice, right? But on the other side of the equation, if you record from invertebrates, animals that don't have a spine, crustaceans like crabs or worms or mollusks, snails, those kinds of animals do not show the same kind of response that we have. They actually show an inverted response.
Yeah.
Okay. So
Yeah, I was very interested in that.
yeah. So it suggests that they are not conscious, at least not conscious all the time. Right. So they may be momentarily conscious, and we are trying to test for those possibilities. Right. So in other words, we can use the device to go up and down the evolutionary ladder and look at all the organisms and find out which organisms are conscious and which may
So
not. Yeah,
let me ask you a question.
sure.
Did you test on any plants?
Yes, we did. And we see an inverted response with plants, but much slower inverted response than invertebrates.
But it's still, the inverted response is not the flat line that you get if nothing is there.
Yeah. So the flat line you get only with inert objects. That inert objects at room temperature give you flat line, like a stone or a rock or some piece of plastic or piece of wood, dead wood.
Yeah.
And so if you get an inverted response, it means there is some kind of a default activity going on. And so we went along to test the idea that, so we've dug deeper into the mechanism now. And so one of the ideas that came out of Anne Rose's idea, and Stuart Hameroff contributed to this, and now a lot of other people who have collaborated with Stuart have contributed to this idea, which is that the benzene rings, ring structures, benzene rings have many loose electrons, what are called delocalized electrons. And so the collapsing electron waves they propose is going on within these benzene rings. And the benzene rings are side chains of some amino acids, which are part of the protein molecule that constitutes microtubules, tubulin, alpha and beta tubulin. So they have what are called tryptophans, phenylalanines, and tyrosines. These are amino acids that are arranged in many proteins, but probably in microtubules, in tubulin molecule, when it has a particular structure, spiral structure.
Yeah.
And that these, especially tryptophans, are positioned at specific intervals from each other. So they form a three-dimensional structure that may be important in amplifying this phenomenon, as opposed to loose benzene molecules floating around or something. So they have to be arranged in a particular structure in order to produce this phenomenon. This is what we are thinking right now. And yeah, you had a question?
Oh, I'm dying to ask you. So you finish and then I'll ask.
So anyway, so we thought, why don't we see if we get a default response? And mind you, the default response is what we get with invertebrates, right? Which is inverted. We also get an inverted response, by the way, if we euthanize a mouse. So in other words, we kill a mouse humanely and
Yes.
properly and all that, and we measure from its head instead of the body. We, for a while, for up to 5 hours or so, we get an inverted response from the dead brain. The dead brain that does not have electrical activity like our live brain, but you get a default response, which then goes away, right? And so the hypothesis is that dead brain still has microtubules. Before the structure falls apart, they still have a structure, and they still have these benzene rings in tryptophan amino acids within these alpha and beta tubulins arranged in this microtubular form. And so they produce, the hypothesis is, do they produce, we asked, are they responsible for this default response, the inverted response with our sentiometer? And so in order to test that, we said, okay, what if we had an artificial material that had benzene rings arranged in some kind of a structure, right? Would that material produce an inverted response? So what we did is, there is an artificial material that has very many benzene rings arranged in a polymeric form, like chains of molecules. And that material is polystyrene. Okay, what you get in styrofoam, right? But you get polystyrene plastic, and the polystyrene plastic is used to make petri dishes for lab experiments. Okay, so we recorded from a petri dish using the sentiometer if we had a petri, we have a petri dish that does not have anything, it's completely empty, and we record with a sentiometer, you don't get anything. It's inert. You get a flat line. But if you put a little bit of water in that petri dish, right, all of a sudden you get an inverted response. Okay, which means water somehow interacts with the benzene rings in polystyrene. Polystyrene is it's a polymer of styrene. Styrene is a modified form of benzene. Probably the delocalized electrons in these benzene rings of polystyrene that stick out within the petri dish, they interact, those electrons interact with the hydrogen ions inside water. Hydrogen ions are basically protons, okay, because hydrogen has one proton in its nucleus and then one electron. So the proton probably gets attracted electrically to the electron, and somehow that produces this inverted response. We have a more detailed, again, mechanism by which that might happen, but it's getting into it would be too complicated.
Well, this is just more nice proof for what Stuart says about how many scientists complain that the brain is too warm and wet to hold a quantum process. So seeing water come in to make some function happen with respect to whatever you're doing to study consciousness is fantastic, because of course many of us believe that water is absolutely crucial to the whole process. I'm dying to ask you about measuring from the hand, because I know from your presentation that you're not only using your miniature double slit device, or now it's just measuring the diffraction, but that you get similar readings from a person's hand.
Yes. So the idea, it comes from what I told you before. The process, the conscious moments or conscious experience occurs, as if Roger Penrose and Stuart are right, in these big macromolecules inside cells in the brain. But according to my extension of that hypothesis, it spreads, that influence spreads through the brain, but not just the brain, it also spreads to the rest of the body, right? Just as it spreads out in space where we measure it, right? And it falls off with distance, by the way. So if you measure far away, like 2-3 feet away, or even a foot away, you don't get much of a response, right? You have to be very close to your head. And
close is close.
Like 5 centimeters. Within 5 centimeters, you get a good response, right? But in the body, it spreads better than outside the body in air. And we have done experiments to show this, right? It spreads better in body tissues, in brain tissue and in body tissues. And so if you are 1 centimeter away from your hand, because it has spread to your hand, you still get the same kind of response that you get from the brain. A little bit smaller, that's all. So,
but this is fantastic.
Yeah. So that is the interesting thing, because we could potentially make a device that you can wear as a watch or an armband or even a ring, if you miniaturize it further, to record the level and content of your consciousness. We are hoping that we will be able to do this in the near future. So
and what I can guess, but what is the most, let's say, likely practical use in the near future?
First, there is a whole bunch of medical applications which require a lot more work. Just barely scratch the surface here. And other people have to replicate our findings. It's just our lab at the moment, my lab at the moment, that's doing this. Although other people are trying to do this and have some success, I have heard quite a bit of success actually. But so the simplest thing you can think of, without getting into the medical applications, would be make a wearable device, wearable sentiometer that measures the level of consciousness in meditators. So while you meditate, you should see some change that is consistent. And depending on the type of meditation, you would see different changes in the level. So you could know how well you're meditating for you by just looking at the recordings. You could just get some sense of how stressed you are mentally, especially if you're worrying about something, if you're relaxed versus you're anxious or you're agitated, whether your mood is depressed or elevated, or all those kinds of things, without going into these ultimately would have medical uses, but those require proper clinical trials.
Yeah, of course.
And would be potentially be useful in diagnosis of some mental illness.
Or a brain injury or whatever, I can imagine.
And brain injury, level of consciousness in the ICU, general anesthesia, depth of general anesthesia, all
That's a
those
big
things
thing.
are, yes. So all
People
of those
occasionally
things,
speak about being like immobilized but not anesthetized, so they can't speak.
exactly. Right. So if you're conscious, and you should be able to detect the fact that you're conscious using this device. It is sensitive to mental activity, so it would be sensitive to different contents, like you said, and that is a whole big area that needs to be explored, and it cannot be done overnight. So it would take decades for us to figure that out if it is the case. So yeah, but the first few kind of tentative steps that we would take is in the direction of using it to look at, to study level of consciousness or level of activity in meditators versus mindfulness, relaxation, level of mental stress, all that kind of thing. Yeah.
Oh, very interesting. Have you had an opportunity? I don't recall from your presentation. Have you had the opportunity to try this device on someone in a coma patient?
Yes, yes. So that's what we are doing in the ICU. So in the intensive care unit, we have patients that are the patients we recorded from. One of them was in a deep coma, and we saw almost no response in that patient. But the others were in a lower, what we call persistent vegetative state or minimally conscious state. So we saw reduced amplitude of the response in those conditions. But this, what we are doing is a pilot study. So it has to be done more thoroughly in a properly controlled, randomized clinical trial in order to really establish the fact that we are able to classify people into different state and look at it from as the patient progresses, whether, and see if it has prognostic value, whether we can tell by looking at the recording whether the patient has the potential to recover full consciousness versus patient that would not recover. And that would require long-term studies.
Yeah, of course.
Of things, right? So
Of course.
yeah, but it's all possible because we have the device, and anybody who is interested, any physician who is interested, who is specializing in those kinds of areas can collaborate with us, and we are happy to collaborate. We already have people who are willing to do that. It would be interesting from all those standpoints. Also, people who we can probably know definitively whether a person is brain dead or not. So this would be a very objective measure of brain death, because if what we see in mice occurs in people as well, we would see an inverted response in a brain dead person as opposed to an upright response that we see within a conscious, even an unconscious person.
And even on the more optimistic side, to be able to see some evidence of a spark of something that's still there that would encourage the medical team to put all their effort, or encourage a family to not lose hope for somebody's recovery, because we can now see that there's something going on inside that we had no way to measure before. That is incredible.
Thank you. In that vein, we actually have patients who show waxing and waning of their consciousness level,
Right.
okay, which was predicted by people doing these studies using other methods, but they were not able to directly measure that. And so for the first time, we are able to measure, like if a person becomes conscious momentarily and then lapses into unconsciousness, we can tell that. Yeah. Also during sleep, we see, although this is still ongoing and we don't know for sure, we see oscillations that people, we don't know if they're dreaming or not, but dreaming is a state of consciousness, right? So in people who are asleep, we see these oscillations that it goes up and down and up and down in a periodic fashion, which we think corresponds to dreaming and dreamless sleep. Sleep is unconsciousness, and dreaming is actually a state of consciousness. And that is what you would expect through the night. So spatial.
I really loved how incredibly thorough your research was. You did everything, like including testing this whole apparatus inside a Faraday cage and using a like placebo version of the device so that a person wouldn't know if they were wearing the real one or not. Like, you covered everything. It was like so incredibly thorough, and
Thank you.
I really enjoyed listening to it and appreciating that this is the birth, I think, of something really significant to our understanding of consciousness.
Yeah. The thing, other area that might be interesting in a more broader sense is whether AI can become conscious, right? Whether AI machines, the way they are right now, can ever become conscious. And so if we are right, we meaning Penrose, Hameroff, and all the people that are in this area, and then us with the Sentiometer, if we are right, consciousness is a biological phenomenon, and we know what this underlying biophysical-chemical basis of consciousness it is important. What the physical-chemical basis of consciousness is, right? So if we are right, then AI can never be conscious as it is conceived at the moment, right? Even a quantum computer, if it's based on standard quantum processing qubits, the way they're thinking at the moment, that kind of a computer can never be conscious. You would need to build a biologically inspired machine that incorporates these ideas, so you would need benzene rings structures in there, structured in particular ways in order to, which evolution has done, by the way. Evolution has made structures like the microtubules or some other macromolecule, right, with particular structural dispositions for the benzene rings to be arranged in particular ways in order to amplify this phenomenon. You would need to replicate that in the computer, right? And make that the way it processes information, as opposed to the standard ways that, using neural networks or quantum neural networks. So it's a very straightforward answer to the question whether AI, as it is conceived right now, can be conscious or not. And the answer would be no, categorically. So the importance of, or the significance of this line of research is pretty tremendous in that sense.
Absolutely.
Yeah.
And just before I let you go, I want to touch on the other really significant inventions that you're responsible for. Tell us about the Oncomagnetic device.
Yeah. So again, it was the continuation of my previous device, which was a transcranial rotating permanent magnet stimulator. It was a TRIP-MS, we call it, which was a neuromodulation device, which basically stimulates the brain and potentially stimulates neurons and causes what we call neuroplasticity. So changes in neuronal connections and potentially useful in many diseases. And it's all in the experimental phases right now. So that same device then was extended further. We discovered that if we modified the patterns of stimulation, the orientations of the magnetic distributions of magnetic field, and also made the device stronger in terms of the magnetic field that it puts out. We could kill cancer cells, especially
Oh,
cancer cells of the brain, right? So the most dreaded cancer of the brain is called glioblastoma.
yeah.
And so we took patients' cancer cells, glioblastoma cells, patients who had glioblastoma in a dish, and then stimulated them using this particular patterns of magnetic field that we had devised, and found that we were killing these cells by just short periods of stimulation. And the way it kills is we rotate the magnetic field at certain frequencies and with certain timings. And turned out that we are not using the same stimulation protocol. We do not kill normal cells, normal
Uh-huh.
neurons, and normal glial cells in the brain, or normal any other body cell, the ones that we have tested. And so it was selective to only cancer cells. And the reason we found out was because of the exact mechanism by which this is happening. As I told you, it has to do with electrons, just like in the case of migratory birds, right? So these electrons have what's called a quantum property called spin. Okay,
Spin.
so yeah, spin. It's actually, the electron is not spinning, but it's got an intrinsic angular momentum. That's what its technical term is, and it's called the spin. And electrons come in two different kinds of spin that are in opposite direction, so up and down. And so normally they are, when they pair up in any kind of chemical reaction, you have to have one electron up. They always work in pairs, right?
Uh-huh.
So one electron in a pair, one electron is up, the other electron is down. But if you have a magnet bring a magnet close by, a high field strength magnet, and you rotate it at particular speeds, especially, or even as a static magnetic field, what you do is uh, both of them, if you have unpaired electrons, they all orient in one direction. Their spin orients only in one direction that aligns with the magnetic axis of
Okay.
the magnet. So you don't get this up and down. You get either up or down
Okay.
in different orientation. And these, when you have it in that manner, they cannot undergo a chemical reaction, right? So they fly off. These unpaired electrons fly off. And if that happens mostly in energy storehouses inside cells called mitochondria. Okay. And there's oxygen being used by the mitochondria in the chemical processes that go on within them. That oxygen that you have inside mitochondria combines with these electrons that fly off and form what are called reactive oxygen species. And these reactive oxygen species are very toxic to the cell. And they produce all kinds of reaction. They oxidize almost everything inside the cell. These reactive oxygen species oxidize the proteins, the DNA, the lipids, and cause immediate cell death. The cell explodes and dies, and also can stop cancer cells from dividing. So the cancer cell stops dividing because of this. The reason this happens mostly in cancer cells is because, to begin with, cancer cells have high levels of these reactive oxygen species. Okay. And by doing this magnetic stimulation, we jack up the reactive oxygen
Wow.
species even higher, which causes the toxic effects. That in normal cells, the level of reactive oxygen species is not high enough. It's very low. right? Because normal cells have good defense mechanisms to quench these reactive
Ah,
oxygen species, to get rid of them. Like they produce antioxidants inside the cells to get rid of these
got it.
things, right? Which cancer cells are not very good at producing. So their antioxidant mechanisms are not that, I mean, are not working well. So that's why they cannot handle this high level of reactive oxygen species. So now that device we have tested obviously in cells, in animals, in mice that are models of glioblastoma. We've also tested them in other cancers like breast cancer, pancreatic cancer, lung cancer, and it seems to be working in all those
Wow.
cancers.
Wow.
Yeah. And there is a company now that has come up to commercialize this technology to conduct patient trials. We conducted some treatments in patients who are end stage patients who have no hope of there's no if the glioblastoma is a very dreaded disease, there's some treatment initially, but that once you do the treatment and the tumor which invariably recurs. Once the tumor recurs, there's no treatment for it. And then patients usually don't survive. The median survival is around 20 months, 16 to 20 months. So they die. And one particular type of glioblastoma called unmethylated, patients die with the median survival is like 9 to 10 months or so. So we have treated such patients when in the end stage because FDA allows us to offer compassionate treatment to such patients, and we have found that we were able to prolong their life, shrink
Ah,
the tumor. Yeah, of course, we cannot cure the disease in these cases, but we were definitely able to prolong life in most of them.
and it's so non-invasive
Yeah.
compared with radiation and chemotherapy.
Yeah, and it doesn't produce any side effects that we know.
Wow.
Right. The patients wear, for glioblastoma, they wear a helmet and they get treated 3 times a day, 2 hours. They have to stimulate themselves for 2 hours each time, 3 times a day. And then they have to keep doing it. And we found that it prolongs life in these patients, including some really even more serious, devastating condition called diffuse midline glioma, which affects young kids at the age of 7. It also occurs in young adults. So we treated young adults who have this condition and we were able to prolong their life as well.
Oh, that's so
And
beautiful.
yeah, so yeah, we are hoping it'll come out. They'll do a big trial, pivotal trial that would potentially lead to FDA approval, and then hopefully it'll come out as a treatment for people with these
Man.
conditions. Yeah.
Yeah, such big things. Have you got another big project on the back burner, or is there a big goal in your life that you would love to achieve somehow?
The consciousness thing is the biggest thing I can do. If we are able to show that we are indeed measuring consciousness and that the mechanism of consciousness is what we think it is, making sure we do rigorous scientific tests and confirm that's the mechanism. It would also lead to some new developments in physics, because if the mechanism that I talked about works the way we think it works, then that would involve new physics. And I talked to physicists at the Barcelona conferences, and they all thought that was the case. So, the first step is to make sure that all of my results are reproducible in someone else's lab.
Yeah.
More people are able to reproduce them and do more variations of the experiments and look at it from many different directions. And that's what my effort will be now. We are trying to put together something which we're not ready to announce at the moment, but a network of researchers involved in trying to use the device to make it useful to people. So if that happens, that's my goal, basically. There's nothing else I can ask. That is big enough for me.
Wow. Yeah, I was blown away with
Thank
your
you.
presentation, and this is so interesting to hear. And I just love it that all of this is coming out of your creative thinking and your
Thank you.
your tinkering and looking for. It's not that it's a low tech, because of course I couldn't build this thing myself, but it feels like a very, I want to say, a simple kind of answer. It's a simple kind of test that
Yes.
is leading to something extraordinarily profound, and that
Yes.
really lights me up.
Thank you.
So
Thank you so
thank
much.
you
Yeah.
so much for doing this interview.
Yeah.
I really appreciate it. If people are hearing this and would like to get in contact with you, of course, I'll put all your information in the show notes, but I guess you're on LinkedIn. Is there another place for them to find you?
Our institutional websites, you can Google me and you'll get all of that. So it's easy. My email, professional email, is easily accessible.
Okay, great.
So yeah. I have a Twitter account, but I'm not very active. Yeah.
I hope that one day you'll be very active with some exciting news. You'll be tweeting it all over the place.
Thank you. I don't have a YouTube channel for sure, but I have those YouTube videos that, the science of consciousness. I presented this work at different stages, the past couple of science of consciousness.
That's actually a really good idea. You can, by Googling you can find the presentations themselves on YouTube.
Yes.
And it's
Yeah.
really worth watching
Yeah.
to see to see all these graphs and all the research results. It was fantastically, I want to say entertaining. It was entertaining, but really, really meaningful. So thank you so much for being here.
Same here. Thanks for doing this. Yeah.
Okay.